All right. Hey. This is annoying Professor Dax, so let's focus up now for our last few minutes of class. All right. So sorry about the technological issues. Hopefully somebody fixes that or fixes me, one of the two. All right, Ajax, did you send that email to IT for me? All right, perfect. Super stoked already for my class after this. So let's continue talking about epidemiology and kind of the things that they look at and how that kind of dictates or drives public health. So hopefully when you see some of these details, you can see how this is incredibly relevant. I guess I don't want to mute that one, whatever. This is pretty relevant to what we're experiencing right now. So most pathogens cannot survive for a long time outside of a host. As you guys have probably heard or maybe read, you know, there's been a lot of information that's come out. I haven't checked a recent update about, you know, how long the COVID-19 virus, right, SARS-CoV-2 can survive on the surfaces of things like food products at the grocery store, right, is a concern, right? We wipe off our desks after class because that is a concern, all right? And so some pathogens are relatively sensitive, right? Don't survive long outside of a host. Some pathogens can survive much longer, right? It just kind of depends. When we talk about reservoirs, right, we are, right, looking at what is maintaining that pathogen, right? What is it? What is that pathogen living in, right? Where is it coming from when it causes an infection? If you see three types of reservoirs, right? Animal reservoirs, again, pretty relevant, and we'll kind of mention that here in another slide in a minute when it comes to COVID-19, right? Human carriers, right? Now, for the most part, we are not great reservoirs, right? We're kind of the terminal end for a lot of infections, so we consider ourselves more carriers, not necessarily a reservoir, although as a carrier, right, you can still infect. And then non-living reservoirs, and we'll talk about those here in just a second. Now, when it comes to animal reservoirs, where we get really concerned with this idea of zoonosis, right? So if you've heard of zoonotic, right, pathogens, this is a pathogen, right, that spreads from an animal to a human. All right. Typically, right, direct contact with the animals or its waste. Eating said animals, right, can be a way that pathogens are spread. And then blood-sucking arthropods, right? So, you know, certain insects, right, can pass pathogens on to humans. When it comes to COVID-19, right, I'm sure some of you heard that we think the epicenter is the Wuhan wet markets in China. You can kind of Google those and take a look. They have a bunch of different animals from all over the world, right? One of the things that's somewhat concerning is there's a lot of illegal animal trade and illegal animal consumption that goes on, a lot of exotic animals in these markets, along with common animals. They're put in really tight kind of caging, stacked on top of each other. And so there's a lot of exposure amongst those animals to one another, right? And then at this market, right, they're also slaughtered and then sold at that market. And so there's a lot of opportunity for animal-to-animal interaction and then human-to-animal interaction. And this is where we think the jump was made, right? So if you've heard of SARS, right, that came out early 2000s, so SARS-CoV-1, right, another COVID virus, also, right, we think spread to humans at one of these wet markets, right? And so just a lot of contact with animals that you might not normally have. Those of you that are interested in HIV, right, we actually think that one of the possibilities is that locals were slaughtering, and by slaughtering I mean not like decimating, but like a butcher slaughters, right, a cow or a pig, slaughtering chimpanzees for meat, and we think that that's how maybe HIV made the jump from primate to man, right? So not uncommon, and can cause a lot of issues. We also, right, with influenza, we have this animal to human jump, right? And as I mentioned, right, we're usually kind of a dead end host, and these are difficult to eradicate, right? We come in contact with animals for food. We come in contact with animals for as our pets, and so this is just kind of something that we're kind of dealing with. Yes. Dead end host. It means, again, that how I said, like, we're like a carrier. We're not necessarily a reservoir, and so we're not walking around with these pathogens and kind of living, I don't want to say symbiotic relationship with them. We're either fighting them off and getting rid of them from our bodies, or they're killing us. All right, they're not hanging out in us, kind of living in harmony. Does that make sense? Yeah. So we're terminal one way or the other. We get rid of it, or it gets rid of us. All right. These reservoirs, right? They kind of have we'll say, a mutualistic relationship. This is a chart from your textbook that just shows some common zoonotic diseases. And some of you have probably heard of these, right? If any of you have had ringworm, right, or you've had pets that have had ringworm, right? It's actually a fungal disease. You can get that from your dog or your cat, right? There's other places you can get it, but it's passed to us from animals. One of my favorite, right, is Lyme disease. I just think it's super interesting. Maybe one of you will do that for your disease report. And so you see a lot of different things on here that you recognize. And so I think you can certainly start to appreciate, right, the role that animals play in passing disease to us. All right.
Hey, Dr. Jack.
Yeah?
Okay, on that table, should we memorize, like, get to know that, like, what type different things are, like fungal, viral, anything like that?
Oh, no. I'm not as bent on that as maybe if in your head you can think of what are a couple common zoonotic diseases.
Okay, awesome. Thank you.
I don't care if you try to go bacterial, viral, fungal, but if I say zoonotic disease, I want you to be like, oh, ringworm. Oh, Lyme disease.
Gotcha.
Okay, good question. All right. So, this is kind of related to Alanis's question, right? So human carriers, right? What can be scary, right, about human carriers, and again, this is directly related to COVID, is that a lot of times human carriers can be asymptomatic, right? So they're not showing signs. And this is one of the things that's kind of up in the air a little bit about COVID. So you guys, for the most part, and kids that are younger than you, appear to be great asymptomatic carriers for the SARS-CoV-2 virus. Now, what's still kind of up for debate, and I haven't checked the literature here recently because it's moving so quick, and I doubt anything settled on this, right? It appears that research is showing that people that are asymptomatic, at least kind of later in the stage of the virus, are not very good at spreading it, right? Which is a good thing. But early on in infection, they are. So there's that. I don't know, right? Some individuals, right, easily develop sickness while others don't. Again, we see that going on right now. I said most of you are asymptomatic, but you might know somebody, or maybe you've recently had it and you've experienced some of the symptoms. I know of a couple former students that had recently graduated that are just a little older than you, were in good shape, and couldn't get out of bed, for a week type of thing from this. While others are like, yeah, I had a little bit of a headache. I took some ibuprofen and I was good, right? It's kind of all over the place.
I have a question.
Yep.
And like you said, this is, all a bunch of information still coming in. But Do you know of any lasting effects? You've heard of anything? 'Cause I heard of some things, but from the reading you've done.
Well, I mean, there certainly is concern already that there may be some lasting pulmonary and cardiac effects that we're already seeing in some patients. But even that, I haven't read to see, are they 100% sure that's permanent? Because right now we're saying something's a lasting effect and the infection's been going on for 6 months. I'm not saying that it will be. It's just, it's so fresh right now. And that's, I think, one of the concerns. Like, I love college football. And I'm a Big Ten fan, so we've already thrown in the towel, which I don't think is necessarily a bad idea. And one of the concerns is, from a liability standpoint for some of these conferences, is if Zach played football, he got COVID, 10 years later, he has some breathing pulmonary issues. And then he says, well, it's because I got this, right? We just don't know. So yes, Zach, we think there are, but again, it's probably going to be years before we really have a good grasp on this. That's a good question.
Which team in the Big Ten?
Iowa.
Ohio State.
Oh. Do you remember a couple of years ago when we ended your national championship aspirations, when we pounded you? Yeah, forgot about that. Okay, good. And this is what, kind of related but unrelated to what Zach was talking about. This is what's fascinating, right? Healthy carriers, right, may have a defense system that protects them. And so what we don't know yet is that probably there is some gene, right? There's some maybe difference in the ACE2 receptor. If you've done any reading on the literature, it's the ACE2 receptor that the virus binds to. Maybe there's a different expression of this receptor in people that are asymptomatic or protected. Maybe there's a mutation, right, where the receptor is shaped slightly differently. There's a whole host of things that it could be, pun intended, see that with the host of things like that, but that's good, right? We just don't know yet, all right? To Zach's point of lasting effects, it's going to be a little while before the research can catch up and explain, right? So people that, I can almost guarantee, I feel pretty confident, when you have someone that's completely normal and healthy, and they get a severe bout of COVID-19 where they're hospitalized or perhaps they even die, right? We're like, there was nothing wrong with them. Unfortunately, probably months, years from now, we probably will find out, hey, those individuals that got it that seemed healthy were actually susceptible because, right? And so there's just a lot that we don't know. So if you're non-living reservoirs, right, soil, water, food, we could even argue, right, if you didn't clean your table and the virus was there, right, it's a temporary spot that the virus can kind of hang out for a little bit, and it allows transmission to occur. Now, truth be told, right, it typically, right, is fecal contamination of these sources that leads to the spread of disease. Some of you that are involved in mission work, a lot of places, one of the big things that's a target is actually access to clean water, right? If you've been to other parts of the world, right, and you've noticed that maybe people are bathing or gathering water downstream of where animals are bathing and doing things in water, right? This is a huge issue throughout history, right? It's been fecal contamination of our water systems that has caused some pretty big outbreaks of different diseases, right? So, if you've ever been camping, right? One of the things that I was taught is don't camp at you can camp at the bottom of the hill, but don't go to the bathroom at the top of the hill, all right? And I know that sounds kind of crazy, but throughout history, it's actually when big armies have moved around, a lot of them have had huge issues with disease because they didn't do their business away from where they're doing their business, right? You got me? Blair, do you have a question?
Yes. So if it's fecal matter from animals, would that be considered zoonosis or non-living reservoir?
Ooh, yeah, that kind of gets you kind of into an interesting middle ground. I would consider that non-living reservoir because that's not direct contact with the contaminant.
Okay.
All right. But I mean, you could certainly, if there was some type of essay question related to this, you could kind of talk your way around that, and I wouldn't ding you for, as long as you kind of explained,
Wow.
hey, yeah. So this is one of my old slides, but these are just showing some reservoirs and carriers, right? You know, one of the big things I think that we don't realize in the United States is one of the reasons, and maybe you think it's expensive, but in the grand scheme of the history of meat prices, it's actually relatively cheap. And one of the reasons that meat is relatively cheap is because we crowd a lot of animals together. When we crowd a lot of animals together, right, we have problems with certain diseases, right? We are great carriers. Again, this is kind of relevant. This is an old slide, but this could be a slide of a COVID party from foolish college students, which I know none of you will hopefully be. Good. Again, soil is a reservoir, right? When we say we have issues with these things, you can't clean dirt, all right? And so this is just a constant source. And again, we don't appreciate sometimes how big of a issue insects can be. All right. Oh yeah, this is one of my favorites. This is our prairie dog. I don't know if you've heard, but prairie dogs are actually a great source, a great reservoir for plague. And so I don't know if it's happened recently, but every once in a while, some person gets the idea of, I'm going to go out and catch a prairie dog and sell them to people as pets. And then some people get the plague, right? So if your dad or mom gave you a prairie dog for your birthday, kindly say no. Right? Again, we've gone over these, right? Vector, any living thing that can transmit disease. Right? For those non-living reservoirs, another word that you may see them called is a fomite. All right. You know, and a great example of fomites, or something that's a fomite for those of you that are going into healthcare, right? If you've seen a patient that's really, really sick, and maybe you're not properly garbed up, right? Well, then your lab coat, maybe if you don't change it, or your tie that you're wearing, right, can become a fomite, right? The virus, the bacteria, whatever pathogen could get on it, you go and see your next patient, and you spread it, right? If you look throughout the history of biological warfare, right, one of the not so bright moments in American history was one of the things that we did to help thin out the Native American population is when we were marching them along the Trail of Tears, right? It was winter for some of that march. The Native Americans were very cold. And so what we did was we had some soldiers in the army that were sick with smallpox. We had them wrap themselves in blankets, and then we gave those blankets to the Native Americans, right? And that blanket, right, was a fomite. It helped transmit the disease. It was non-living, okay? So that is a historical example, and again, not one of our, I think, proudest moments as a country. All right, so epidemiology and public health, right? One of the things, right, that they're kind of looking at or focusing on is disease transmission. And when we talk about disease transmission, which we kind of have been hitting at in various forms here, you have to look at two kind of main arms of this, right? You have to look at the host, and you have to look at the pathogen, right? So some of the factors that we care about in the host. Immunity, right? We're talking about those people that are maybe asymptomatic or don't even get infected, right? What is different about them, right? Some of you may be naturally immune for whatever reason, and that actually kind of gets in genetics. We can talk about an example of that in just a second, right? Is there herd immunity present in the population? Has an individual host been immunized? What's herd immunity? We'll actually talk about kind of how you roughly calculate this when we get to the vaccine unit. What's herd immunity? Okay. Yes, that's getting closer. That's part of the puzzle, right? So you've exposed a lot of people, right? And they are immune.
So, with most people that don't get sick.
Okay. Yeah. So we have a lot of people that got infected. They built up immunity. They're not getting sick. So what's the, Alana, finish it off. Close.
I was gonna say it's like that everyone gets vaccinated.
Right, exactly. So the idea of herd immunity is that a large enough percentage of the population has become infected and has recovered, right, that the pathogen is no longer able to spread amongst that community. One of the reasons that vaccinations are so important, right, protecting from certain diseases, is that you have members of the population that can't get vaccinated for a variety of reasons. And we'll talk about that in a little bit. So by you getting vaccinated, right, if enough people get vaccinated, enough or a large percentage of the population is protected, those that can't get vaccinated, right, are then also protected, right? So it's kind of a benefit for them. When we talk about, you know, COVID, the idea, you know, will probably be at some point is either through natural infection, which if we go back to Zach's point, we actually are still not sure how long the antibodies last from a prior infection to SARS-CoV-2. If you look at SARS-CoV-1, right, the OG, those folks had immunity that was maybe not perfect. It was present for maybe 2 to 3 years after. All right, so, but again, that's a related virus, but we just don't know, right? You can't tell me right now that someone that had COVID-19 is going to be fine a year from now because we haven't been able to measure their antibody responses a year from now. Okay? But one of the things that herd immunity will probably do, for the most part, right, for SARS-CoV-2 by COVID-19 is protect the elderly and protect folks with underlying conditions. Again, general health. This is like a storybook kind of layout of SARS-CoV-2. Again, I showed you that hospitalization data, and we saw that people that have hypertension, high blood pressure, people that are obese, have other metabolic disorders, are more prone to disease. And that's not just COVID-19, right? That's in general. Age. Well, you know, right? Older people are typically more susceptible to certain pathogens. We start to get really concerned as a population with pathogens that are actually, well, I mean, we get concerned when they start killing anybody, but right, a really bad pathogen is one that can kill, you know, young and old. Gender, if we actually go back and look at that SARS-CoV-2, there actually is a little bit of a discrepancy in infections and hospitalizations and gender. I have to go back and double check that. Males are actually slightly more susceptible. Again, we don't quite know why yet. Culture makes a big difference. How you live, right? Maybe how your family unit is structured makes a difference. A lot of different things. Genetics, right? Why is there more of a protection against malaria in people that are from Africa or of African descent? What genetic disorder do people in that population have that actually protects them from malaria? Sickle cell, right? And so what sickle cell does to a red blood cell makes it not as an attractive as a host for malaria parasite. All right. Genetics also, people that are of European descent sometimes have a mutation in a cytokine receptor called CCR5. All right. Does anyone know what that protects from? HIV, right? And what's really fascinating, do you know what other disease it protects from, Michael? So what's really interesting is that that same mutation actually protects from smallpox. And so the idea is, many, many years ago when smallpox was an issue, right, it wiped out a larger portion of the population that didn't have that mutation, right? The folks that survived, a lot of them had that mutation, and then they passed it on, right? Now, that mutation, all right, is not common in people of African descent. And so what we see right now is that on the continent of Africa, there's a much higher susceptibility to HIV than there is in Europe and the United States, right? Because we have that mutation present in our population and it's not present in the African population. Yes?
Can you go over a quick definition of your herd immunity again?
Yeah, so herd immunity is the idea that a large enough portion of the population is now immune to a disease that it protects those that are susceptible. All right? And so you get to herd immunity two ways, either via natural infection or via vaccination. All right? Again, I'm kind of curious, are we going to get close to that with natural infection before the vaccines even out? I don't know. We can talk about that when we talk about how you calculate the percentage of the population. So different different pathogens have different numbers for herd immunity. And kind of the quick rundown of how you do it is you calculate that based on the number of people that one infected individual can infect. Right? So again, I'll show you how that works. So for like the herd immunity, the magic number for measles is actually, I want to say, I think it's like 95%. It's really high because one person with measles is able to infect a lot of other people, right? It's highly contagious. So the more contagious the disease is, right, the higher portion of the population you're going to need. Right, so for a pathogen virulence dose in incubation, we'll talk about virulence in just a second. Dose, I don't know if you've heard, but there's some speculation. If we talk about COVID-19 as an example, like let's say I have it and I walk by Morgan and I don't have my mask on and she breathes in some of the air, but we keep on kind of going opposite directions, right? That's a low dose, a relatively low exposure. And so we think right now that she would have very mild symptoms, right? We've seen some healthcare workers, regardless of what physical shape they're in, right? When they're in a hospital with a lot of COVID patients, there have been a number of them that have died. And we think that's because of constant exposure, right? A higher dose of the pathogen there, right? Incubation time, right, plays a huge role. How quickly does it multiply within a host? At what point during that incubation is it infectious? That varies from different pathogens. Sometimes it's only when they're showing signs and symptoms, right? What gets tricky with a lot of pathogens is there might be a period right before they get sick, right, that they don't show signs and symptoms, and that's when they can pass it. There's a lot of different things that are kind of in play there. So when it comes to virulence factors, right, one of the words that we have to focus on is pathogenicity, right? The ability of a microorganism to cause disease, right? And it's able to do that because of this virulence factor. And it might not be something that you necessarily think about when it comes to causing disease. So what are we concerned about, right? Certain adhesion factors. What's an adhesion factor going to allow or do? But don't overthink it. Yeah, how does it stick on? How does it attach, to something? We'll talk about later in the semester, and I'm kind of sprinkling it into here. Certain bacteria have something on them called capsules. Capsules are kind of a mucousy, sticky shell, right? They might allow that bacteria to stick onto something that they couldn't have before, right? Biofilms, right? Who can give me an example of a biofilm that we should be concerned about? Huh? Yes, plaque. Yes, please. Brush your teeth. All right. Streptococcus mutans, right? Likes to form biofilms in your mouth. I'll show you a picture of biofilms later. On a microscopic level, they are disgusting. All right. When you see them, you think, oh, that's on my teeth. Maybe I should brush them more. Right? And biofilms, we'll talk about these, are great at protecting pathogens. They're also great at allowing different pathogens to live in that biofilm and sharing information. So if there's a pathogen in there that's really, really smart and knows how to maybe resist a drug, right? That could be an issue. If any of you have had MRSA right before in your life, right? That's formed of methicillin-resistant Staph aureus, right? And the biofilms that they form there, right, make it very hard to treat. Right, extracellular enzymes, right? So the ability to release something and maybe break down a target to gain entry. Toxins. So some of you have probably had the DTaP vaccine, right? You're getting vaccinated two of the things, diphtheria and tetanus. In that case, those bacteria themselves don't actually really do that much too. It's the toxins that they release that cause a lot of issues, right? And then anti-phagocytic factors. What does that mean? What's phagocytosis? Savannah, help me out. Right, they eat up the pathogen, right? Or they eat up the damaged tissue. And we'll talk about next unit. I just want to have it put in there, right? That certain pathogens have gotten really smart and can either actually sometimes use some of these toxins to kill phagocytes. They can hide from phagocytes. Some of them can even survive within phagocytes. All right. And so these are all different sorts of things that contribute to the virulence of a pathogen. And so I'll let you go on this slide. So one of the things that's awesome about public health and epidemiology is the data and information that they gather, right? They are looking to break the chain of infection. So you have your reservoir. You have how does it leave that reservoir, right? That moment of transmission, where does it enter, and who's susceptible? And so the idea would be, with the knowledge that's gathered from epidemiology, right, working with public health, if we can interrupt this chain at any point, right, then we save or protect that host. Right? And so I'm going to let you go now. What we're going to do starting on Wednesday is talk about some of these portals of entry, some of these portals of exit, and then we'll talk about some of the actual kind of groups of studies that epidemiologists do to help protect individuals. All right? Get out of here. I hope Zoom works for us on Wednesday. If not, blame Holmes. All right? It's been good. I'll see if I can get the kinks worked out for you guys later. Thank you, Ajax.